Good evening, everyone. Welcome to virtual shadowing session 103. Thank you all for continuing to show up each and every week. Tonight, our guest is Professor Karen Babayan, and she will be sharing an artistic approach to medicine. Next slide, please. Here is our working group composed of Taylor, Rohit, Kiana, Elena, Cheyenne, Ali, myself, Christina, Ashita, and our 4 physician leaders, Dr. Fowler, Dr. Salazar, Dr. Wynn, and Dr. Reno. Next slide, please. Our upcoming sessions next week, session 104, we will have an interesting talk about what makes us human, and it'll be about exploring brain cells one at a time. The week after that is still to be determined. On 6/28, we'll have a specialty spotlight in pathology, followed by a PA spotlight on skull-based neurosurgery on the 5th. Next slide, please. As for the quiz following tonight's session, we will share a link to the 10-question multiple choice quiz, both by QR code and in the chat at the very end of the session. When you pass with a 70% or higher, you'll be prompted to download a certificate. That serves as your attendance and participation. You will have 1 week to complete this quiz. As an extra incentive for staying till the very end of our live session, we will reopen an old session, session 27, and that link will also be shared in the chat. It will not have a due date also. Next slide, please. For our Q&A, typically we have 2 Q&A sessions, one at the middle and one at the end, but today's presentation is quite unique and has many different components. So we'll have a few Q&A sessions spread throughout, and we'll stop for a few questions after each. Dr. Fowler, would you like to say a few words?
Yes, thank you, Aditya, and welcome to Virtual Shadowing. We have a really exciting presenter tonight, and as you know, along the way, we've tried to present different angles in looking at careers in medicine, nursing, physician assistant, and so forth. And so this is yet another of those marvelous examples of someone who's become amazingly well trained in 2 different crafts and has become a communicator to be able to use the visual elements, in this case, artistry, to be able to lead a path to understanding very complicated subjects. We appreciate you coming week after week. It means a great deal to us. I can't believe we're at lecture 103 already and into our 3rd year of doing this. And so we just want to thank you for coming. Please, when you take the exams online, there will be a GoFundMe, and if you can send $1 or $2, we'd appreciate it. It all goes for the supporting the costs of the cost for the Zoom, the cost for the exam software, and so forth. So anything you can do would be sincerely appreciated. Again, thank you for coming. It's so nice to see so many faces again, week after week. It means a great deal. You've become an extended family for us, and we just want to thank you for coming. So Aditya, do you want to introduce our marvelous guest for the evening?
Yes, sir. Without further ado, it is my pleasure to introduce Professor Karen Babayan as she showcases her artistic ability. This will be an interactive lesson, so please grab a pen and paper so that you can draw along as she teaches us about body systems and how impactful art has been in the history of science. Professor Babayan, please take it away.
Thank you, Lydia. Hi, everyone. So I'm going to do something a little different. It's not different for me, but it's probably different for you. And it is for artists and non-artists alike. So it doesn't matter if you can draw. I'm just going to put that out there. And that is not the point of it. The point is the experience. And I want to actually, make this so that you have a bunch of experiences, not just digging into one particular subject. So this is a sampler. And I would discuss some of these topics in much greater detail. But since I wanted to appeal to a lot of people's interests, I went with these 3 particular topics, and I'm going to link them together. So my goal is that we have a big, vast umbrella topics like evolution and ecology. We have organismic perspectives. We have population perspectives. We have organ system perspectives. We have microbial perspectives. We have genomic perspectives. And since I'm in a genomics lab right now, I focused a little more on genomic perspective, but I was originally teaching general biology and anatomy. And I love that broad perspective. And I think that we can weave in and out of these, and there's multiple layers, and you create your own universe of experiences, and that all galvanizes into your point of view about the phenomenon of living things. So what I have here in this little picture here, I did write a little graphic novel on epigenetics, and I used 2 characters in it. And we're going to see them later. This guy Raj and his cat Socrates, who's actually my cat. But the nucleus. So I'm just going to do things a little differently. You may not have seen biology done as a storybook or anatomy done that way, but that we're going to do a little drawing after each topic. There's 3 topics. The first one is the cystic fibrosis gene, the CFTR gene, and the disease cystic fibrosis. I'm going to look at that. And the second topic is going to be the microbiome, which I find to be absolutely fascinating, just because it's a vast universe that influences us in ways that our brain can't really, fathom right now. There's just so much going on, but it's such a cool topic because of the way it influences everything from our behavior to our skin. And that's a cool topic to visualize and to think about. And our last topic is going to be from my own shadow experience, where I followed a reconstructive surgeon around because I wanted to teach anatomy to my students in a unique way. And so I developed a graphic novel about the thyroidectomy, a total thyroidectomy. So at each one of those, I'm going to stop and we're going to actually draw something that goes with this, something on a board. I usually would stand in front of a class with a big board, and I'd move in and out, back and forth. And we're going to do something similar here so you can get a feel for just the idea of breaking down something, the contemplative nature of drawing. It is a very contemplative process. The overlap between drawing and surgery, which is really cool because it's eye, hand, and brain, and the self-efficacy of feeling like you have a skill and that you're developing a skill and you can self-teach yourself and you can share your knowledge with other people. So let's start with a little bit of the illustrated history. And there's just so many people in history that have contributed immensely to the life sciences that get scant recognition for their artistic skills. And everyone knows Leonardo. Leonardo's life is beyond interesting. You know, I'd be remiss if I didn't tell you, look him up, read his notebooks, his philosophy. He's really, really a powerful person in history because of his passion for nature. And that's really where it stems from. And his discussions on the heart valves, but it ties in with things you wouldn't think are related, like the flow of water in rivers and the vortices that go around when currents are created. His notebooks are worth reading. They're a little hard because of the translation, but they're worth reading because of the breadth of knowledge in them and his philosophy. So, and there's people from all over the world, every different country, the ancient Sumerians, Akkadians, Egyptians, everybody had science that was really astonishing, and we forget about that. Sir Charles Bell is another personal favorite of mine because his brother and himself were surgeons, but they were engravers, and I have a couple of books of their work. He's best known for Bell's palsy and the cranial nerve that he, established in that condition. But his work is phenomenal to look at, and I highly recommend it. And this, of course, is Theodore Bovary. Most people don't know Theodore Bovary, but he's actually a guy that him and his wife, Marcella, resurrected Mendelian, the Mendelian ideas of Gregor Mendel and genetics. They actually established the probability as an actual thing that happened. But he is the father of cytology. And around the 19th century, he started out as a landscape artist. And that's what he wanted to be, a landscape artist. Did not want to study zoology, but he ended up doing it because his father wanted him to. And he illustrated chromosomes, the nucleus. This is the beginning of the genome. Really, it starts with him and his phenomenal understanding of chromosomes as individuals. And if you read his work, he established the idea of chromosome territories, which to this day, people are still trying to comprehend. And this is all from observation. He also posited a theory of tumorigenesis and inflammation and genomic instability, all with a microscope and watercolors and etching. And this is amazing, but most people don't know about it. So if you get a chance to look at some of his beautiful work, I really would suggest you look at it. And this is a good example. Both of these have a merger between the thought processes of art, how they culminate in with this, how they sort of merge with the scientific processes, and you culminate into a theory, and you culminate into an experimental procedure or something you can test. And it's really amazing. So Bovary's fantastic. Then we have Santiago Ramon Cajal, who is the father of neuroscience. Again, a man who did not want to go into science, did not want to go into medicine, wanted to be an artist, but he listened to his parents. His father was very strict. My father did that too. He said, "What are you doing with that art? Don't you do that. Take some science." So, but these people were so passionate with the art. His work is beautiful, and it's very reflective of the Art Nouveau movement and the turn of the century, and almost a lot of Japanese art. It sort of reminds me of that. So, and all of his work was this detailed staining of neurons and dendrites and axons and extensions, fibrous extensions. And this is how he became the father of neuroscience. It was through the art and through the experimentation with it. And Da Vinci did the same thing. He discovered a lot of the way the heart functioned, by making his own model of a heart and playing with it until he understood it better. So this is a really beautiful picture here, and you can kind of see that everything looks like a neuron. Everything he draws is a neuron. Every pattern is a neuron. So his work is definitely worth checking out. So we've got the father of neuroscience, father of cytology, surgery, general natural sciences, anatomy over here. And then we have Cushing here, notably well known for his pituitary gland surgery. And I'm most astonished by the technique that he used, which was called carbon dusting. And carbon dusting is a technique where you actually scrape carbon. Harvey Cushing did this with a guy named Max Brodel, who was basically the father, I guess, the 19th, 20th century of medical illustration. And they both knew each other's work and topics really well. But the carbon dusting was where you scrape the carbon from a charcoal, a piece of charcoal, and you get a dust and you paint with it by using a beveled brush. And from that, you make this phenomenal looking piece of artwork here. And if you see some of it, it gives an incredible three-dimensional look to stuff. It's anatomy, and it is a very, very skilled technique. So his work, again, worth checking out. And of course, everyone knows Frank Netter, and just because you end up seeing his books everywhere. And I think his style is definitely 1950s style, but so spot on accurate. You can't really, there's no one I know today that replicates that level of detail and can convey both disease and healthy states and all kinds of things that you would need as a medical student. So this is a really brief overview, just so you know. All these people are there for you at any time you want to look into their work, and there's so many others. It's an enormous number of people that you just are too many names to say, but this gives you an idea. So our first topic, genomic medicine visualized, is cystic fibrosis. So most people know cystic fibrosis, and most people know genomic medicine. They've heard of the term. Genomic medicine, just I'll real briefly say, is different from genetics. And genetics is a study of single genes and their effects, but genomics is a term coined maybe 20 years ago. It's not just single genes, but the functions and interactions of genes in the genome. And the genome is an ever evolving, system that is within our nucleus and a little bit in our mitochondria, which has crosstalk with our nucleus, and all of our chromosomes. So all of our chromosomes are our genome. So genomics has a broader, like more ambitious reach now than genetics, and is a lot of experimental, interest. There's a lot of things you can do with it, but it extends beyond genetics to complex diseases like breast cancer, colorectal cancer, HIV or infections, tuberculosis, Parkinson's, Alzheimer's, obesity, diabetes, many multifactorial disorders where you have to explore patterns rather than just the single gene, single protein effect that you get in monogenic diseases. So a lot about expression of genes as well. So I picked cystic fibrosis because it is a so-called one gene, one enzyme disease, but there's tremendous variation within that. And it really is an interesting story. And it's characterized by tremendous variation. So this thing, I used to think this myself, oh, you got a deletion, and now you're going to get cystic fibrosis, and everybody's cystic fibrosis is the same, and it's not. So I would say, oh, you could visualize this easily, but then when I realized that there's this remarkable variability, severity, rate of disease progression, organ involvement, all this stuff is based on the types of mutations, how many mutations, your own modifying genomic system within you, your DNA repair mechanisms, how your chaperones work, your health in general, your diet. So all that goes into influencing a disease that you might think is just everybody's got the same disease. It's certainly not true. So there's a lot of different stuff in here, but we're going to look at some of these things. And these are the primary organ system. I picked 3 antique drawings because they're just incredible. I wanted you to see them. The lungs are mostly associated with cystic fibrosis, which is a chloride ion channel defect. And we're going to look at that more closely, where chloride ions accumulate in the cytoplasm or on the other side of the membrane rather than leaving the membrane. And you get this really, really sort of, I guess, an electrolyte balance. You get this really thick mucus, and you've got epithelial cells all through. Oh, let me fix that. Right about that. You've got epithelial cells all throughout the whole respiratory tract and goblet cells, and you're secreting mucus all over here. So the mucus is going to be thick and viscous in people that have the mutation. And so the lungs are going to be the most dramatically affected. Then you're also going to have the pancreas is going to be affected as well. You're going to get clogging of the pancreatic duct. And a lot of times there were some stories and recordings of people in the 16th century where their pancreas was described and people thought witches had taken them over. And it turned out to be cystic fibrosis. So the pancreas is another organ. The digestive system, of course, has mucus lining throughout it. So that system is affected. And the vas deferens are also in males are affected. And you can get, infertility as a result of that. But these all vary from individual to individual. And that's why genomic medicine is kind of important in that way, because it seeks to personalize medicine so that you can't treat everybody the same. So those are 3 major organs, but we're going to look more at the lungs. So we call it a monogenic disease, and we call that mutation in the CFTR. Boy, the phone's off the hook, sorry. There. So we call that mutation, the actual name of it, CFTR, and the gene encoding the epithelial ion channel that normally transports chloride and bicarbonate ions. And so the mucus is what is developing as a result of a protein that has been misfolded. So the CFTR is the gene, and it makes the protein that will become part of the plasma membrane as a transmembrane channel. So characterized primarily by a pulmonary infection, inflammation, pancreatic, exocrine insufficiency, we said male infertility. And of course, most people that have cystic fibrosis don't live past 50. Some people have children, some people don't live past 20 or 30. A lot of, in more severe cases, you need a heart-lung transplant, but once you get it, these genes, the defective genes just keep working and they keep making the abnormal mucus. So this is an autosomal recessive disease. It's most prevalent in Northern Europeans. 1 in 20 are carriers. And there's some interesting historical perspectives about how it may have come about, and that it may in fact be a balanced polymorphism and may have some heterozygote advantage. But there are many CFTR mutations. So here are the normal lungs of a person that doesn't have this. And here are a cystic fibrosis person. You can see there's a lot of scarring in their lungs because over time, it's just kind of like, I guess, similar to emphysema, or, because the alveoli break open, the mucus will cause lots of infections. Bacteria will get trapped in there. You'll get repeated respiratory infections, and ultimately your lungs won't work or function normally anymore. So this is kind of like cystic fibrosis was linked to the Black Plague. It was linked to tuberculosis. It's linked to cholera. It's linked to syphilis. A lot of people have suggested that one of the advantages of having this mutation was it prevented these types of infectious diseases. But this is largely conjecture. But it's interesting because there are documented cases where they've discussed this, and it turns out to be cystic fibrosis in medieval Europe, where there were certainly a large number of plagues. And I always think this is a kind of interesting character here, because he's got this little bird mask on, and they used to stuff herbs in here to help them, protect them against infectious agents. They thought by wearing that, they'd be able to do that. So the mutation may be very old. Some people say 3,000 years. Some people say 40,000 years, but did it benefit anyone really? We don't really know. So why am I telling you all this history? Well, when you're going to think about the narrative or the story of a disease or a condition or a mutation, you have to have multiple perspectives. I were going to write a story about this and illustrate it. I would want to get as many points of view as possible. I'd want a historical one or an anthropological one. I'd want a genomic one. I'd want an evolutionary one, and I'd want a strong anatomical one. Then I could piece together, an explanation that is visual for myself. And part of making a storybook is doing that. You're actually manufacturing a hypothesis, and you're making it visual, and you're reflecting on it through the creation of your graphic novel, your comic book, or your story. So all these things I have to take into account if I'm going to make something like that. So these are just some of the general symptoms of it. I might include this in my storybook. I might include this if it's going to educate, the mass public. I might include it if I'm going to certainly talk to my genomic medical students. And I would include it if I talk to my general biology students. So just show you a couple things. And of course, I put the skin in here too, cross section of it, because sweat glands will produce lots and lots of salty sweat. And when babies, they would actually taste their sweat and it'd be really salty. That's how they would test for cystic fibrosis. So here's the standard autosomal recessive, pedigree here. And if you have little c, you're normal because it's autosomal recessive. You have big C, little c, you're a carrier. And if you have big C, you're normal. So autosomal recessive conditions are pretty rare. Autosomal dominant ones are even more rare, or else they kill off the population. And nature is really amazing. What it does is it purges deleterious genes out of the system. And that some of them get left over, but and this might be one of them. It may have a heterozygote advantage, just like sickle cell. We're not quite sure yet. So where does this happen in the genome? It happens on chromosome 7. And you can see there's a CFTR gene in red that they're pointing to. So that's the actual gene. And you take a look at this and you see the protein, the primary structure of the protein. And there's a lot of ways this can go. If you remember protein synthesis, which you could draw with your picture, if You could draw that with your picture. You'd have protein synthesis, and you could get a misfolded protein. You could get no protein at all. You get a truncated protein. And all of those will determine how severe your cystic fibrosis is. That's why it's always good to illustrate these sequences of biological cellular activity. And here's the most common mutation. It's F508. It's a deletion, and phenylalanine gets deleted here, and you get an altered sequence, and then you get an expression of the disease, which is manifests into a phenotype that is disease causing or doesn't work. So it's very interesting to try and imagine what a mutation event looks like. So whenever I look at a book and I see something like this picture, where they talk about it, and they go, well, we had a translocation, or we had an indel, or something happened, where we lost some part of the chromosome. You want to visually imagine what's actually going on in the architecture of the nucleus among the chromosomes that would result in these kinds of mutations. And that's a really different world. It's a very abstract thing to imagine, but it's good to imagine it because it makes you think more deeply about it. So we got the valine in there. Put that there. So here's another view, and we always think of this as there's the gene. It looks like this. Is that really what a gene looks like? We don't know. But we make a schematic of it so that we can understand it better, and you can see the spectrum of mutations that would affect the function. You've got in-frame deletions, missense mutations, nonsense mutations, frame shift mutations, splicing mutations, all kinds of different things. Then you've got the cellular level, which you juggle with the molecular and the genomic, and the cells that'll be affected, in this case, the goblet cell and the respiratory epithelium. And I just thought I'd show you this picture so that when we draw, that you think about what cell this is happening in. And so here's just a little histology piece. And slime is a big part of life. And I think this is a really cool thing to do. I would look at hagfishes because they make a lot of slime. When you spool DNA from doing, extracting DNA from strawberries, which is a very common experiment in general biology, you see it as this slimy, snotty looking stuff. And that is the stuff of life. And knowing those things is really important. So, because that gives you a feeling for what mucus does, and if it gets too thick, what it would do to these cells and what it would do in the lungs. So here's like a airway that's normal, and here's an airway full of mucus. And here is the plasma membrane with the chloride ion channel that we're going to draw, and the mutant version of it over here. And of course, alveoli are really the functional components of the lungs, because there where gas exchange happens. So these are going to all become clogged up with mucus. So we'll put something like this in our illustration too, so that we have a perspective of the anatomical with what's happening in the plasma membrane. So this is a really great drawing, and I like this because what they did was they gave you wild type cell or normal cell on one side, and the pathology of the cystic fibrosis cell on the other side. And you could compare the chloride ion channel in each part of the membrane, and you can see what happens from the time the chromosome is, makes its mRNA, and the mRNA leaves the nucleus, and the mRNA is transcribed and translated by tRNA and ribosomes to the dysfunctional protein that eventually gets incorporated into the membrane. So it's really something like this is also good to draw because you get to see intercellular dynamics, and you get to review protein synthesis, and you get misfolded proteins. And I always think of misfolded proteins kind of like origami that hasn't gone well. And when they don't fold well, you get a lot of disease. And then the body's always trying to get rid of misfolded proteins, but in some cases it can't if the gene is there. I mean, it's the expression of the gene can change, but the gene is always there making them. So of course, mentioned before, this has got a lot of variation in this disease. There's almost 2,000 mutations to date that have been detected. And the heterogeneity of clinical manifestations in individuals with the same CFTR genotypes definitely indicates disease severity is modulated by other genes. So there are modifying factors in cystic fibrosis, and that's a big challenge, you know, because we don't know the modifying factors. There's many of them. They may be variably expressed at different times. Diet may influence them tremendously. We don't know. So this makes this simple, straightforward autosomal recessive disease really complicated. So I guess we can start calling complex disease complex disease now, rather than just complex disease. So I'm gonna stop there, and before we do, you know, I want to think about, how do you think transmembrane proteins evolved? And this, for this thought, you want to take yourself back in evolutionary time, in deep time, you know, 3.5 billion years ago, and you've got protocells, and you've got you know, amphipathic membranes coming together, and you know, anybody has any thoughts what would happen to transmembrane protein? How would it get in here? What would it do? How would it start this whole you know, selectivity thing going on? So, and I just wonder myself, you know, because we can't go back in time, but what event triggered these selectivity portals that made the plasma membrane a really, really, you know, amazing communication system, a protective system, and one that basically controlled what the cell is going to experience. So this is a really hard thing to visualize. You just have to gather data, and then you have to think about it from multiple sources, and it's just something to get your mind to think. So I'm gonna stop sharing here. Let me see, where's my stop share? Let's get this out of the way. And we're gonna go to a new share. We're gonna look at get your paper and pencil out and everything, and see, and get to my little board here. And what we are going to draw is a real easy thing. So if you're somebody that doesn't like to draw, this is going to be so easy. You'll just go, wow, that was really easy. And since I usually sketch things, we're going to put our plasma membrane right here, and we're just going to make this little two curved lines right here. We're just going to make two curved lines going over here. And we're imagining that we're looking into the cell. We're looking into a part of the cell. And down here, we're going to make another curved line. And that's just going to be where our nucleus is. So we remember that this membrane is part of a cell. And that's part of the epithelial membrane lining the respiratory system. So then we're going to put a location where we're going to put our chloride ion channel. I'm just going to draw a circle in there, because this is how you would lay out a drawing. And I find that drawing the plasma membrane with its component features is really, really great way to study, because there's so many conditions that affect membranes and things within them that if you draw and redraw it over and over, that's great. And we're going to put in the corner here an alveoli, and we're going to make a little cylinder to show that this is a terminal bronchial. And then we're going to make some circles here to indicate that these are alveolar sacs. And we'll make them like right here. And then I've got a layout now here, and you do too. And all I did was draw some circles and some lines. That's all I did. I didn't do anything special. Now I'm going to go in and make it a little more solid. And usually a way to make yourself feel anchored with a drawing is to do some contour lines outside of it. So we know this is the plasma membrane, and I'm going to give you an easy. We're going to draw the little, you know, like the hydrophobic, hydrophilic parts of it facing the outer body fluid and the part facing the cytoplasm. So I'm just making little circles here, because these are going to represent those pieces. They're going to represent, and once you get used to drawing a little bit of circles and cylinders, you can draw just about anything. So here we're just representing them. So we can sketch in those hydrophilic heads. You remember from old biology, some hydrophilic heads. And this gives us the landscape that we're exploring the CFTR mutation within. So we'll just do this, and you know, you can color. This is great if you make these in black and white. You can color them in later yourself and make your own coloring pages. And if I had more time, I'd show you a real dynamic drawing where we could take sort of a Marvel Comics perspective. We're not going to do that. So we'll put our hydrophobic fatty acids in here, and then we start thinking about this as a membrane. And we've already got our subject sort of laid out. Takes a little time for people, but it's just a wavy line. Remember, it's just that, and it's just these, and they're all in here. And we have cholesterols in here, and you can put that in here, and we can put a peripheral protein on here if we want. We can give this some skewed perspective if we want. But for now, we've just got a membrane, and that's great. We know the nucleus is down here, and we can just put some nuclear pores. And just a little trick, when you're trying to create the illusion of a sphere, your circles become more skewed. As you get towards the center, the circles become more rounded. And that gives you the feeling that you've got some three-dimensionality to your nucleus. So we've got that. We know what's going on there. We know the genome is inside of here. And then we're going to put our diagrammatic CFTR, our chloride ion channel in there. So we're going to draw this, and we put a circle here, but it's got it's a protein. So it's not just a round, smooth thing. It's got a lot of like indentations, kind of reminds you of like a rock. And if you look at a rock, sometimes crumple like paper, if you crumple some paper up, you get these domains, you can sort of envision them. And then you can sort of see there's some other binding domains below this, and you can just sort of, these again are just curved sort of cylinders, but they give you a feeling of what this might look like. So when you see it, you can feel like, oh, I can alter this now. So, and of course, we were to look at this, we'd see that this membrane goes over the cell. So it's contiguous through the cell. So we put a few little indications that it is going over, and our field of view is going over the cell. And then we can sharpen that up a little bit. We're inside the cell here, and we can sharpen our protein up a little bit. And I can make this a little bigger. We can get a feel for this and make it a little bigger. So we can accentuate it. And you can see this is a pore here, so the chloride ions go in and out. And if you were studying this, you might say, oh, maybe I'll include some other things. And then we'll do the same for our alveoli down here. We'll make them kind of get that, look like there's little alveoli inside of them by giving them a little bit of a texture on the outside. And this gives us a feeling for we know what's going to happen, because this is a cylinder here, the air is coming in here and going out here, and there's smooth muscle on it. And these are all little sacs, individual sacs. And we know that this might be covered in that viscous mucus, and it's going to ultimately have a bad impact on the function of the alveoli. So we'll draw that in there. I'm going real fast. I shouldn't, but I don't want to make it, because normally I would say go slow with it. Since I'm just giving you a little sampler, and we'll do a contour line on the outside, and we'll get a feeling for those alveolar sacs. We know. And of course, you've got capillaries going all over these things. So there's capillaries, and you can draw them in red and show them all over here. And you've got your membrane here, and you can put the chloride ions down here if you want to write them. They're all building up here in the defective channel. In the normal channel, they would move right through that pore. But here they get built up. They can't get through. And sometimes in really bad cases of cystic fibrosis, you don't even get you get no protein at all. So you get no protein. But we can draw the mRNA coming out of here. And remember, there is the endoplasmic reticulum here, which is going to process this. So we can draw that. And again, these are just lines. There's nothing spectacular about what I'm doing. Very straightforward. And you can draw that. And then, of course, the Golgi complex is going to package it and pinch it off and ship our channel that we've made, the channel protein, to the membrane where it'll be incorporated. So if you do something like this, and it is like really easy, you can see that it's an easy thing to actually draw. And they can do this for any, if you were studying, I don't know, tetrodotoxin, or you were looking at, sodium channels and potassium channels, you could do the same thing, and other transmembrane proteins and portals. And of course, there'd be an aquaporin here, so we could put an aquaporin, which pulls water through. And because this thing erases, I can erase this, and I can put an aquaporin here. And water is also coming in and out of the cell. And if you make this page for yourself, and you put CFTR gene, chloride ion, cystic fibrosis, this is something you can color later on. And if you want to put a chromosome in, you can put that on a board. I would draw this whole thing out. It'd be much bigger. And we draw chromosome with it, and we draw we draw the gene and everything, and maybe the lungs too. So, and one thing I learned from Da Vinci is he used to put these lines behind everything, and you can do that as well. It kind of makes your piece of art that you drew pop out. So that gives some background and shading to it. And now you've got it. So
Corinne, and Karen, this is Ray. Can you save that? And I'd like you to send it to me.
I'll send you the original though. I can send you, I did it on paper. I'll send you that one. It's better.
Oh, that's cool. That's cool.
This stinks. So, but, but I just wanted you to get an idea for it. So, and I can pull up some things that I did. So I'm going to show you those in the other one. If I can find them. There's plenty of artwork here.
Yeah, Karen, a bunch of our students have put in chat that they will pay for a signed copy. So
okay. So I wanted to show you what I did for my students here. I have it somewhere, cystic fibrosis somewhere in here. We did HIV, we did cystic fibrosis. This I just did actually for an article that will be coming out in American Biology Teacher. And this is just the steps of viral invasion. And as I mentioned to you before about playing with crayons, this was done in crayon and colored pencil paper. And this is like just so much fun. I got to play with this. I really enjoyed the perspective. And that's what I wanted to show you here. I have a different point of view from like across the surface of a bacterial cell. And you've got this phage attaching here. And then you're looking at it from that point of view. So you can do stuff like this panning out and looking at this bacteria here with the little phages. Then you can pan in, and then you can do a storyboard. So it's great to do those things and a lot of fun. So I'm going to stop this one. And I guess if there's anybody has a question, I'd be happy to answer it. And we'll go back out to our PowerPoint so that we can do our second.
Yeah, there are a few questions. I'll give you one or two, and then we can move on to the next one. Do you think that providing high quality medical drawings could play a beneficial role in the physician-patient encounter? And if so, how do you envision integrating it into a medical visit?
I think it can make a huge difference. I think in medicine, the closer it is to reality, the better. But if you come across a disease that is molecular or genomic, then you're going to have a little, you can make some abstractions, you can verbally explain it. I do think it would make a big difference. I certainly think surgeons, plastic surgeons, reconstructive surgeons, when they want to explain to a patient what they think they might look like, that's really, really useful. I think your ability to be able to illustrate this is what's happening, or even a metastasis of a cancer. You're showing somebody, hey, look, this is your large intestine, and this is where the polyp was. And I think and you're drawing it and you're saying, well, I think it metastasized to your liver, and I think it through your lymph nodes made it down here and did this. I think it is extremely beneficial. And I also think the flip side of that is, I think people that can illustrate and illustrate things getting better is actually cathartic, mentally cathartic to do. So yeah, it can play a lot of different roles, not just communication, but really, I think some kind of therapeutic role for the patient, very much so. Absolutely.
So one more question. How do you take such a complex medical condition and turn it into something that a general audience can perceive as a graphic novel or friendly story? What is the creative process like, and what inspires you to take on a particular topic?
Oh, wow, that's a good question, because there's so many components to it. It is a manifold experience. But like, let's say, I tried, like epigenetics was really interesting to me because nobody can really put their finger on it. And there are so many things happening. But at the same time, even the metamorphosis of a butterfly is fascinating. And I'll say, well, how do I illustrate a metamorphosis? Well, you'd say that's easy. You show the caterpillar, you show the imaginal disc, you show the chrysalis, and then you show the butterfly. But it's way more than that. So I would think, wow, how can I put this perspective in the genomic perspective? How can I put an evolutionary perspective in here? And then these pieces all fit together in a story. And the other really pivotal point of making a good story is having characters to help you tell it. And so I think I'm probably going to answer that question when we get to the microbiome, so you can see a part of it. But yeah, you have to feel like you are the character. So I think taking on the character, these are points of entry into any phenomena. You have to have multiple points of entry, and you really have to be able to adapt to what would it be like if I were there, or this was me, because nobody's down at the molecular level. Nobody sees what these things look like. They don't even know if they have color. So it's really interesting. So I hope that answers that question for you. We'll probably answer more of that later when we get
Definitely. In. Please share your screen and feel free to carry on.
Okay, thank you. Okay, so where are we? Okay, so the microbiome is another topic that I've been interested in this for many years, because I knew there was something else going on. You can't we are really beings of this planet, and despite the fact that we think we're so separated, and soil becomes us, it becomes through the fruit and vegetables and everything grows, and it gets into us, the nutrients, everything is becoming who we are. And I always tell students this, I say, where do you think the phosphate in your DNA comes from? And then people think, wonder where that comes from. Do we eat it? Yeah, so you eat it, it comes from the erosion of rocks and minerals and everything. So it's all components of you, are components of this planet. And I think if we become a healthy substrate by the kind of things we eat and do, then our microbiome becomes healthy. But there's so much to learn about this. The fact that you can, this is when you're in the womb, you don't have much of a microbiome, or when you come out though, your microbiome becomes this really complicated thing that becomes part of you. And now you are going to be inoculated by your mother, and then you're going to touch different things. And we forget that it isn't just us that has a microbiome. Cats and dogs and raccoons have a microbiome. Plants have their own microbiome. So we're in constant contact with these systems that are being transferred to us. And the microbiome has been implicated in so many diseases, because we have a microbiome on the surface of our skin, in our eyelashes, in our armpits, in our groin, and certainly in our gut, and all these other areas of our body. And there are all these little ecological niches that these microorganisms inhabit. It isn't just bacteria, it's many, many different types of species. So this is really complex biology, and it's hard for us to wrap our mind around it. So I took a narrative approach to this because I thought I would tell a story about the microbiome, and that way I could read lots of different articles, form my own ideas about it, and start to try and develop a multi-level story where we go in and out of this system and we get to explore this system from different points of view. So yes, the gut is the main one. A lot of like chronic inflammatory diseases of the gut, and inflammation of the gut, and autoimmune diseases, of course, and even behavior are linked to microbiome dysregulation. Of course, we have lots of these organisms in our oral cavity. I think that's where we have some of the greatest diversity, that in our gut. They influence brain and behavior. There's a lot of crosstalk. Their genome and our genome are essentially in many ways one, but some of them are not that integrated and some of them are. So they're called these an axes, where you have brain, skin, and gut connected together. They're overlapping. The evolution's happening inside of you all the time. So they're evolving, they're competing, they're out-competing, all that stuff. So the earth and all kinds of interesting facts become who we are. And that's what the microbiome is in its very simplest way. So here's just some numbers for you that you can look at. 10 to 100 trillion is the number of symbiotic microbial cells in each person. And most of them inhabit our gut, which is understandable. That's where the nutrients go. So we're creating the environment they're going to be in. So the food you eat will create that environment. We'll talk a little bit about that later on. There's 10,000, which is like another number I came across, the number of different microbe species that we've been able to identify. 90% of all the disease that we think, might be traced back to the gut. This is really amazing. I was reading an article the other day that your skin microbiome will determine whether or not your wound will heal well or not. So that changes a lot of things, in terms of the way we think about stuff. And of course, we're killing off a lot of our microbes all the time, and we have to strike a balance between what do we disinfect and what do we leave alone. For a long time there was that hygiene theory that like people are scrubbing things too much, using too many antimicrobials, and they're actually causing the evolution of antibiotic resistant strains. And so the factory farming industry does that all the time because they're constantly giving antibiotics to those animals. They don't live in good conditions, all kinds of stuff. And I read this was interesting about factory farmed fish. They're overwhelmingly contaminated with antibiotics, antifungals, heavy metals, all kinds of things now. So all this, we're affecting these systems in very dramatic ways. So 22,000 is the approximate number of genes that we have. And that's just our genes, but 3.3 million is the number of non-redundant genes you might find in the microbiome of the gut. Really interesting statistics here. And they tried culturing them, but it's very hard to culture some of these and identify them. So they've used metagenomic sequencing and made a lot of progress in that, especially in the intestinal microbiome. And they study dysbiosis with this, but they also study evolution, and they study novel functional genes, microbial pathways, and we get to see antibiotic resistance genes, which in nature, if you put pressure on something, you're going to get variants of it. So 10 times as many microbial cells in the human body as there are human cells. And this was based on, I think, very conservative estimates. They don't know absolutely for sure. You tend to make more of them or get more of them as you grow older. And maybe they diminish. I haven't read anything on aging yet, but I'm going to look that up later. So we said that already, number of microbial species identified. 91% of the cells in our body are non-human microbial cells. So are we a body, or are we a bipedal, ecosystem walking around, with all these things happening in us, and eyelash mites and viruses living here and there and dormant, and these microbes in different places. I think looking at ourselves as an ecosystem is a great thing, because succession is happening on us, and each microbial fingerprint is unique to each person, and that's in each individual. So wow, you look at that, microbiome contributes about 99% of the genetic material that encodes the proteins in our body. So there's a lot of crosstalk, and there is a lot of interaction, and there's a lot of gene expression that has been, sort of structured by these microbes in our body as well. So the composition varies by anatomical site. Among mammals, the microbiota is extensively conserved. The stability determines the major enterotypes, the colon, vagina, mouth, and skin. Of course, dietary changes can alter it, and the way you were inoculated at birth, whether it was cesarean or whether it was live birth, that changes the type of microbiome you have as well. Seasons, temperature, just like an ecosystem, it changes it. And of course, drugs can dramatically alter the composition, and age alters the composition as well. I just don't know what happens to you when you get really old with this, but you're like an old island, and these things are inhabiting you in an old island. So just here's some breakdown of the kinds of bacteria, and you can see some of them here. You might have seen some of these on your yogurt container, like lactobacillus, staphylococcus, streptococcus, firmicutes, and fusobacteria, proteobacteria. And so mouth and tonsils, and some of the diseases like cavities and gum disease and gingivitis, and the placental microbiome, of course, and the vaginal microbiome. And this affects yeast infections, because if you have a reduction in good microbes and you get a proliferation of yeast, then you have a problem that's hard to get rid of. And that's usually from somebody taking antibiotics for a very long period of time, and they can't restore their gut microbiome or their vaginal microbiome very well. And of course, skin and diseases like psoriasis are somehow also linked to this. And of course, they do a lot of studies with the gut and the stool. So they use stool samples to categorize and do metagenomics and sequencing of the different species of bacteria. So some of the terms you might see with the microbiome or biofilms, and we're actually going to look at this because I thought this was a really interesting topic to illustrate. So I'm going to show you that. Quorum sensing, which is another really bacteria talking to each other, exchanging information, communicating. They're very social. And this has really changed dramatically. 20 years ago, no one would say bacteria are social. No one said, oh yeah, bacteria like talking to each other and communicating with each other. But they do. And then they identify fungi and they identify viruses, and all kinds of stuff is happening. So they are extremely social. You don't need a brain to do that. We've got gene expression. That's another word you'll see. And you'll see the 16 small subunit rRNA, and you'll see the Human Microbiome Project. And you'll see these ecological terms like commensalist, mutualist, symbiosis, parasite. And you can go back and forth. You can be a mutualist and then become a commensalist, or usually you work by becoming a commensalist and then a mutualist. You might be a benefit at some point. You might be a problem at some point. So there's a lot of ecological concepts and evolutionary ones that it's good to know, because that's a good way to think about the microbiome: communities, niches, neighborhoods, competition, succession. So here's a 3-domain tree, and Luca down here is our hypothetical last universal common ancestor, 3.5, 3.8 billion years ago. And here are 3 major branches. We have the bacteria, these are prokaryotes. We have the archaea, which also live in us too. They are prokaryotes, but they have a close relationship with the eukarya as well. And in the eukarya, you get the 4 domains, kingdoms, the animals, plants, protozoa, and fungi. So this is really, we're dealing mostly with this. We get some protozoa, we get some fungi living in us. And it makes sense, they all kind of congregate together. So our 3-domain tree is important. And when I went to do my graphic novel of the microbiome, I started with the last universal common ancestor. I wanted to get into that because I thought, wow, that would be an interesting point of entry. I wanted to see if I got the picture here, but I don't. So Luca is really interesting because it's a deep phylogeny, and it made me think, how many genes, how many networks were necessary to sustain a functioning organism? And that's where I started my story. How many genes, how many networks were necessary at this point, 3.67 billion years ago, to make a living cell? So Luca must have had a small number of genes, and it must have had an old type of ribonucleic acid, probably RNA. It had some replicating system, and that changed as it diversified and moved into the present. So that was really, really interesting to me, and that's where I started. So, so here's a metagenomic view of diverse microorganisms in bacteria primarily. There's, I think maybe yeast, no, they're bacteria. So they use 16S RNA gene, which is the most commonly used taxonomic marker to profile microbial communities. They found that over 1,000 common bacterial species in the gut, 3.3 million genes, as we said, and hopefully, if you're healthy, a nice, rich, diverse microbial community. It seems like the less diverse it is, there's even a microbiome of our lungs, and that is another interesting area, because one might think, oh, what are microbes going to do in your lungs? But they are there. So what's their benefit? What do they actually do? So, but our gut, we need a really nice, diverse community to be functionally good. So here's some of the different organisms you'll find. Of course, bacteria are a major focus. You'll find viruses. You'll find fungi, lots of different candidiasis, which is the one that you don't want to get. You'll find protozoa, some parasites like worms, some invertebrates, just kind of, they have some benefits too, and some protein. These are infectious agents, prions, but prions are not as horrible as I thought they were originally, but I'm not going to talk about them tonight. That would be a really interesting topic to illustrate, because they seem to have some adaptive benefit to eukaryotic cells. So another interesting area. So some are commensalist, some are mutualist, and you might think a parasite might be a bad thing, like a tapeworm. It's not a good thing. Nobody wants that. But some worms appear to have some benefit because they're protecting their host. So helminth worms, there's some question about whether they're a frenemy or not. Could they be doing you a benefit? Could they be doing you a harm? So they're actually an emerging source of therapeutics for inflammatory disease because they seem to release certain molecules that modulate the effects of other parasites and other infections. So that's kind of interesting too. And this is, I think the ecological perspective is really useful here. So there's a close-up of somebody. That's a good view. So here's a little few pictures from my graphic novel, Lost City of the Microbiome, which I used to teach because I like telling the story, and I can re-illustrate things when people ask me to. So this is an hourglass of time representing deep evolutionary time and things that went on in it. And it was called Lost City of the Microbiome. And Raj and Socrates were my characters that helped guide me, and I used my research, and they kind of conveyed that to the audience. And so to show you a little bit of the characters. So Raj loves his cat, Socrates, and they actually take you on the adventure through the intestine because they're always studying together. And Raj is a pre-med student, and the cat's always helping him in the book. They have a lot of philosophical discussions, and I use him for a lot of things now. I used him for I started him out with the one on Parkinson's, and I used him for epigenetics, and I used him for the microbiome. So I used him for 3 different books because I really liked them. They had an interesting, I became both characters. It was really fun. So Socrates is always provoking him to think more. So he's kind of, he's fallen asleep and he has too much work and he doesn't know what to do. And the cat is his meditative, philosophical companion. I think cats are that way in general. So that's why he made a really great character. And so he helps him study the topic of the microbiome. And he starts to think about what would it be like to get inside the microbiome. Now, Socrates has very special skills. He has photonic skills. He can travel through light and jump through light. So he takes Raj down into the professor's gut. So they get into the professor's gut through this photonic pathway that he leaps through because of his special cat functioning senses. And once in the gut, they start exploring all of this. And Socrates makes contact with a bacterial cell. So this is where I started the understanding of the relationship between communication and bacteria in quorum sensing and biofilm formation, because now the cat would be communicating with the bacteria and they'd say, oh, what are you going to do next? And looks like he's going to form a biofilm. And quorum sensing is this really unique communication they have. It's sort of a chemotaxic communication or a chemical olfactory, if you want to call it that, communication. So it's really chemical communication to organize the cells, the colony, the population. So then I storyboarded biofilm formation because he's making contact, because what you have happening here is the bacteria will communicate with each other. Some of them will secrete some slimes. There's a gene actually that does this, and then this will build into a home for them, a slimy home. I called this a Slimex community, and this will be one of those things that promotes disease because you're getting these congregating bacteria. So you have, attachment where they attach to a substrate that's irreversible. They secrete these chemicals, they mature, they disperse, and then they leave and disperse, and they go into other areas and they start a new Slimex community. And they use quorum sensing to do this, and that is their communication system where you get cooperative signaling and you get the cells communicating with each other and different cues, chemical manipulations. And this is, their population, and they're having this social dialogue within it, just like you'd have wolves in a pack and you'd have the alpha male communicating to the beta male, and you'd have all kinds of interactions going on here. And inside the gut, this is what would be going on. They're doing this in you right now. They're in there, your diverse community. If you ate your yogurt, you have some extra people coming in there and they're all having conversations. And so you'll get bacterial peptides that will signal the brain that it's full, and they interact with the endocrine system of the gut, or the enteric endocrine system, and they initiate a lot of complex reactions with that. And you'll get some phage transfers of viruses among anaerobic bacteria. There are Peyer's patches, and these are lymphatic structures in the intestine. They're actually visible. You can see them. They're like nodules, and the bacteria live in them, and they live in crypts. So you'll get they have their homes all inside of here in the mucosa, and then you'll get some pathogens will stimulate the immune system and they'll release some toxins. Sugars will be available for them to feed off of. And you may have noticed this, like, if you ever have a yeast infection, you get a craving for sugar. You're like, why do I crave the sugar? Because the yeast are telling you, get me some sugar. And so you'll get the sugar and you'll eat it, and you'll just make the problem worse for yourself, but better for them. So, but there's like all kinds of things going on here. They'll change the composition of their environment. And of course, these microbes do change the composition of things, just like they would change a lake into a salty lake by the metabolites they secrete. So they're influencing their environment, just like we change our environments. They change theirs, and whatever you feed them will sort of direct what they're going to change, or the environment they're going to create, or the species that are going to be left over. And a good example of this is the study where they looked at the gut microbe diversity of humans And they looked at different humans, people in agricultural societies, hunter-gatherers, and people in the US. And I guess you have an easy time of guessing, excuse me, who's actually got the worst gut of anybody, and that's people in the United States. So hunter-gatherer people had the better gut, agricultural people had slightly better, and industrialized countries had the worst gut microbial diversity. And of course, if we looked at chimpanzees and we looked at orangutans and we looked at gorillas, they have very rich gut diversity. Of course, chimpanzees have an actively functioning appendix, so they can break down cellulose. We have a vestigial appendix. So one of the possibilities is that the appendix may have, since it's sort of lymphatic tissue, may have harbored those bacteria for us to break down cellulose. Who knows how many thousands of years ago? And maybe some people still have the genetics for a slightly functioning appendix. I don't know, it's possible. But they found out when you put chimps in human zoos and human countries, their gut bacteria gets bad too. So they end up getting, apparently their diverse food sources are really important in their environment. The soil and everything are inoculating them. So you got a real problem in that low diversity that we have in the gut. Just like if you eat a lot of meat, that changes your gut diversity, and they start producing different metabolites rather than the ones that go with, what happens when you eat vegetation, which changes your microbes. So we've got these different comparisons here, and you can build this phylogeny based on the different species that inhabit the guts of primates. So that's a good way to look at that, view our relationship. Then we've got things like C. difficile, which is a very hard bacteria to get rid of. And difficile infections usually happen after people take antibiotics and they wipe out all of the gut bacteria. If you take them over a prolonged period of time, just like if you keep washing your hands, over and over and over again, when using antimicrobial soap, which has been people, been told not to use, you can just use regular soap, but that's going to increase the resistance genes. So if you've been taking antibiotics for several months, then this crazy thing happens. I gotta unplug the phone, sorry about that. Sorry about that. But so they also kill the good germs, and you get this nice illustration here, which I thought was kind of cool because somebody did this. I didn't do this, but the cells aren't happy in the gut, and there's colitis, and there's plaques, and there's damaged epithelium, and there's toxins that are being, and there's endospores by C. difficile. So there's all kinds of things that are happening. And in this little image down here, histologically, you see 24 hours after exposure to C. difficile, which is now going to be in the feces. 30 hours, it's changing the composition of the lining of the gut, and then 36 hours, it's incredibly inflamed, and some of the cells will burst and die, and the spores will leave the colon in diarrhea, and they may go on to infect somebody else. So this is an example of, a bad bacteria, but something induced by antibiotics. And of course, I did the fecal transplant in my book as well, so in the graphic novel, and they're running away from the fecal transplant, even though the professor is getting this fecal transplant to fix his very difficult bacterial infection. And this is a really old process, goes back a long, long way. You get donor feces, and it's, of course, it's processed. So, they make sure there's no harmful microorganisms in it. And just by inoculating the person's intestine when they have no other choice, it seems to have a really positive benefit. And it seems like people like this have been taking probiotics, trying to inoculate orally by taking the probiotic, and they get no result. and their last resort is a fecal transplant. So that inoculation seems to be very beneficial for people, and they do get them. So, or you can take some capsules, but somehow getting the bacteria right into your intestine seems to really work for cases of C. difficile infection, because there's such a reduced diversity that the communities cannot recover. So I depicted all of this in the book, and I showed you some of this because I wanted you to get a feel for some of the different perspectives you can have, and how you might explore these different topics in the microbiome, because they're very, very useful ideas to kind of think about, especially in health and disease, when they have such a great impact on our bodies. So I'll stop right there. And if anybody, you want to ask yourself, can you see yourself as a unique ecology? Anybody want to answer that? Does anybody see themselves as a unique ecology?
Karen, the study of the microbiome is one of the ways of the future coming in medicine. All the organ interactions that you discussed are so critical. I'm also reminded of what happened in the late 20s when Alexander Fleming discovered penicillin. And while we have saved a lot of lives, we have obliterated a lot of microbiome.
Oh, yeah, definitely. I think that's interesting, because we have a love-hate thing now with them, because we need them, but we're not really, we don't find microbes particularly palatable. And maybe it's changed over generations. Maybe people, since you mentioned the 1920s, people born after several generations are getting a less and less diverse microbiome as they're born. So that's also kind of interesting as well. So what I want to do is, if nobody has any questions, we're gonna draw a, we'll get rid of the other one, and we're actually gonna draw a little bit of the gut microbe in here. Put a new piece in here so we can start. And what I want to draw is, and I have a couple of ideas that I wanted to do with this, but we want to just draw the intestines, and we want to draw a little bit of the gut so we can have our perspective. And so we're going to start with, I'm going to flip this page so I can get a little more vertical. That thing is really wobbly. Nope, stop that. I can't flip it. Okay, we'll just do it this way. So we're going to we'll make where we might see our anterior view of the gut. So just if you make two lines like this, and the easy way to do this is, and we could make a skin cross section too, just to remind ourselves that the microbiome lives on the skin as well. We don't talk about it as much, but I just made a cube here. And the cube is just so that we can fill out the skin and draw from the skin. And then we're going to do a close-up of the Peyer's patches. I wanted to also do those as well. But by doing this, we can say, okay, we're gonna make our diaphragm right around here. So we can separate our thoracic cavity from the rest of our body here. And here we go. And we look at that. We've got a place for our large. We can put the liver in and everything. We don't have to. We can put the liver right under here. Just make a little line here like this. You can do that real easily. And then we can put our stomach right around here. And then again, just make a little curved line around there. And then we're going to make a box where it's going to help us lay out our large intestine. And then we're going to put our small intestine inside of it. So just by doing this little simple drawing, you've got a layout. And if you want to, add somebody like there's arms here, you can do that. You can make that. And then, like I said, with the lines, you can do that last, or you can do it just to give yourself a chance to say, oh, this is what this looks like. And so we just put that there. So we're going to just do the large intestine and the small intestine. And most people see it when they illustrate it. They see the large intestine by looking and seeing these, like the transverse colon, the descending colon, the ascending colon. So we're just going to draw these little lines like this. And I don't like to think about the terms while I'm doing the drawing. I like to think about the shapes. So I kind of leave the terms out of my head until I'm done with the shape. So again, I'm just making these little, these are reference points for my large intestine. And we've got the appendix, which would be kind of here. And then we've got our colon, and just loop this little line around here, right here. And our rectum, which comes down here. So I just put this in here like this. And we're going to illustrate a few of the details of it. And we'll do a couple other things with it. By doing this, I kind of get a feel that this has got fecal matter in it by drawing it this way. It's that it is, going to process it. It's going to absorb water. There's going to be about a pound of bacteria living in here that are making vitamins for us. And I'm just fleshing out what I believe the shape would feel like as it's carrying out that physiology. And then I'll put this in here like this. And we'll put this down here like that. And we'll draw a little bit of the appendix here. And then to give this more of a three-dimensional feeling, there's going to be a shadow along here. So we can make a shadow along here. I think I need this too. This should be just sort of curving with it around here. And there's a shadow here and a shadow here. And then we've got a little bit of it because it's sort of shadowed here. And another shadow here. So this makes us feel like putting a shadow, but this has a three-dimensional feeling to it. And you can easily do that. This is a really quick sketch. It's nice to take your time. And if you're around anatomical models, then I would highly recommend you use them because you can get a lot out of them. And so then we can go back and create a contour line outside of this so that it makes us look more at the inside of it, pulls our attention in there. And go back here, and so we've got this, and you can just kind of get a feel that this is sketching it is good. You get a feel for three-dimensionality that way by doing that. And there's always shadows in here. So this would be your large drawing that you would make if you were going to actually, make multi-step drawings here. And then to just a quick drawing, you'd make the inside small intestine. Of course, that's longer, but the diameter is smaller. So if you just do these lines and indicate, and I'm not really drawing cylinders, but it's curving around, and there's mesentery. We're not going to draw that, but you get a feeling that there's a small intestine in here. It's looping over. And you can go in and highlight some areas and do some spaces here and a little bit here, a little bit there. And you get a feeling of your large and small intestine. Just like for the skin, and you want to draw the microbiome, and you can like say, we'll take a cross section of this, or we'll take a cross section of our small intestine out, and we'll look at that just like your skin. And you can say, here's our 3 layers, and here's my hair follicle, and the sebaceous gland, and there's free nerve endings, and there's piscine corpuscles, and there's dermis, and then you can talk about the microbiome in the skin, and you can talk about it in the intestine. And so one of the things you can do with this is you can take, and that's what I'm going to do right now, just wanted to show you, you could draw that, is we could take, here's a section of our small intestine. We can take a cross section of that, and we can look at the villi, and because they'd all be all in here like this. You can draw this real easy. It's kind of a fun thing, easy, fun thing to draw. And they're sort of waving. And they have lymphatic vessels going into them and capillaries and everything. But we're interested in the Peyer's patches where these bacteria live. And we'll just draw a little bit of that membrane. And then you can draw the cells. You can do it along with me because it's really easy. These are just little simple shapes. And they're all going in here. And it gives you a feeling of what this interior might look like. And I illustrated this in my book because Peyer's patches are immune microbial spaces. They're islands, basically. And bacteria have a nose for them. They migrate towards them and they make homes out of them. They're a great place to live. You know, it's a really nice habitat to be in. And so you'll see it's like a landscape. And when you take this perspective, you've got the Peyer's patch like right here, these crypts, and there'll be a nodule here, and there'll be one here, and you might see one here, and you might see a different type of cells associated with, especially nervous system cells. So you might see a nervous system cell here, an immune system cell, and you'll see all different kinds of things associated with them. And then you'll see the bacteria all along here, and they're going to inhabit these crypts, and you can make them kind of like crypts, make them dark, make them look like they're spaces where microorganisms might go. And so they're going to have an immune system function. We can sort of see them. And then you get a feeling that this is kind of like you can do a thicker contour up here. And you can see, we've got a section of the small intestine. And we feel like we have these little islands here. And you can sort of just fill it in with some darkness to give it that feeling. And make some more cells around it. And this is a great image. I did this one in pen and ink because I really got to do some detail on it. So I'm just making you a real quick sketch here. And of course we can put some extensions on here. So we have a little more detail on those cells. And as I said, you can use stippling techniques, and even doodling works really good. So if you like doodling, because the shapes are not definitive in the sense that we identify them as like an anatomical shapes, they're more cellular and they have a lot more like different ideas, like they're swirling around. And I would give this give the illusion that they're active and there's motion, just like when I did the Slimax community. So here we are. So now you get a little feel of how this and the bacteria are all over here, and they're in here and there, and there's crypts that they're hiding in. So you can randomly put them, and you can make your shapes. And this is what we might illustrate the Peyer's patch as in the intestine. So it's a complex network. There's codependence. It's driven by different metabolites and different pathways, antibiotics, stress. Stress affects the microbiome in very serious ways, and we don't know exactly what the pathways are, but stress makes a lot of sense, seems to make everything worse anyway, for the bacteria as well. And toxins can disrupt this web of metabolic events, and this leads to disease. So we've created an environment where our bacteria can live. So I'll stop that drawing there, and we'll go to our last topic. And let's see, let's stop sharing. We'll go to our last topic here. And let's see, what was I, oh, we're gonna share this screen. So go back here. And our last topic, okay, where are we here? Okay. Is that it? Okay. So everybody can see our last topic. Okay, so our last topic, oh, I got to get rid of this. I can't see it. Hold on a second. I'm not seeing it. Let me reduce this. Okay. So our last topic is going to be the thyroidectomy story. And this actually takes a while to do because it's complicated. I'm certainly no physician or surgeon. And the reason I did this was because I wanted my students to have an experience with anatomy other than a textbook and other than just glazing over the anatomy and memorizing it for a test. So I decided, a friend of mine let me go in and watch him do a bunch of head and neck procedures. And I illustrated while I was doing them. And I came up with this idea because he does a lot of thyroid cancer. And I put together a graphic novel for his residents, but I used it more than he did. And I made it simple enough that my students could enjoy it. Don't do this at home kind of thing. That's not the idea. So we use drawing models and surgery to connect with anatomy. So before I did that, again, I took an evolutionary perspective because I used a phylogenetic tree to talk about the early origins of iodine. And even in the graphic novel, I talked about the 3.5 billion years ago, where you had ultraviolet radiation, and you had iodine-rich seas, and you had cyanobacteria and cells that were sequestering the iodine and maybe using it as protection. And so again, it was born into this colloid that ends up in the follicles of the thyroid gland. And of course, if you look at some other, you'll find it in sponges, thyroxine, you'll find in sponges, you'll find in polychaete worms, you'll find cyclostomes like lampreys, and you'll find them in amphibians. And their main thing is to do differentiation and control metabolism. And in amphibians, they carry out the whole process of metamorphosis. So taking this evolutionary phylogenetic approach early on gave me a broad perspective. And we talked a little bit about where all this comes from. And I always think that's a great perspective to have. So where it comes from. And so I simulated the story adventure of a surgical procedure. I had a lot of fun making the story. It was a time travel story. And I don't want to get into the idea, but the surgeon actually ends up getting turned back into a young kid, and he doesn't remember how to do the thyroid surgery. So he finds the comic book that he wrote, and it teaches him how to do the thyroid surgery, and he relearns it through the comic book, which was my point on making drawing really important. So I used this to learn about head and neck anatomy, introduce evolutionary thinking into anatomy, develop eye-hand skills to better experience anatomical forms, because I built a clay and wooden model of the thyroid too, so I could do this. Develop visual spatial temporal thinking, and make personalized stories about patients. So this is the cover of the book. It's funny how I made this. This hat I made out of paper, so it's not a real hat. And I had some old dental equipment, so I laid it around there. And a young friend of mine posed for me, and I messed around a little bit and got this cover. And this was called Dr. C and the Thyroidectomy, and it was a very graphic novel. And he was telling me, the surgeon, that it's really important to illustrate hands, because people don't see the positions the hand positions you should use to hold the instruments and how you're supposed to approach it. So I used a lot of hand drawings throughout this, and I did a lot of recap of the surgery so you could see the hand over and over again. So this is just one picture I illustrated right here from this, so you could see the thyroid gland from this position. And I wanted, for surgeons, the most important thing was watch out for the recurrent laryngeal nerve and make sure you cauterize everything with this cauterizing scalpel and stuff. And so I tried to highlight those things aside from all the muscles. I was interested in my students understanding that there were these layers of muscles, and that, you have to imagine these layers of muscles, and this glandular thing, which has this huge evolutionary history, with fascia bound to the trachea, and that's a good perspective to have. So we also have the histological perspective and the thyroglobin and the follicular cells and the parafollicular cells that make calcitonin around it. And in that way, I could explain how the thyroid works and the whole control through the hypothalamus and the pituitary gland and all that. It had a nice contextual framework to it. So we talked about T3 and T4, and these are really old hormones, and they go back, they're protein hormones, they're made from tyrosine, they're iodinated, and I believe T4 is like sort of a precursor to T3, and that T3 becomes the active hormone. And the main takeaway idea for mammals is that this is involved in thermoregulation, in our metabolism, and keeping us warm in our metabolic rate, so heart rate and body temperature, and it affects a lot of things. The thyroid gland affects the ovaries, reproduction, the heart. So it wasn't just a gland doing a few little things. It's really, really important. So affects brain development. Of course, if it isn't working early on, you get mental retardation, muscle contraction, skin and bone maintenance, of course, with calcitonin and parathyroid. So there's a lot of genetic variation in thyroid hormone pathway genes. There's a lot of polymorphisms in the thyroid stimulating hormone receptor and in the iodothyronine deiodinases. And of course, in autoimmune diseases like Hashimoto's disease, there are HLA genes which implicate the immune system. So you can also tie in a genomic component to this as well. So I illustrated a lot of the surgery, and I used some of those to make the characters in this have dialogue about what was going on. But I also did the step-by-step of the surgical procedure, so you could see what was going on. So you'd see them talking, and then you see the surgery. And because this book had a Victorian feel about it, I set it in modern Philadelphia, but I also set it back in the late 1800s because I have frequented the Motor Museum in Philadelphia a lot. I gave a couple of lectures there. I don't know if you know about that. That is the oldest, I think, medical museum in the country. And it's phenomenal looking inside. It's a Victorian, beautiful marble and everything. And they have a museum of medical anomalies. And I worked there a long time ago, but it scared me. But they have people with that had problems, like all kinds of skulls with like fangs. They have like a cast of Siamese twinning. They have what they call the soap lady, where she became saponified. They have this guy with this massive intestine, and they have all these historical books that you can go, and they have lectures. They give lectures there. They have a medicinal herb garden, and they have beer parties out there. It is a really cool place to go. So I gave this book of mine a Victorian feeling, that it started out in the Motor Museum, or the College of Physicians and Surgeons in Philadelphia. So there's my young character. So the surgeon is old, but he turns young, and he can't remember how to do the surgery. And he finds the comic book that was written on how to do the surgery using drawing, and that's what helps him do it. But I did a lot of artwork for this so I could understand it better. So I have lots and lots of art pieces, big art pieces, and we actually had a narrative in medicine show at Penn using those art pieces, and we talked about it. So this was like the actual surgery here, so I could highlight the hand positions and specific things that happened and the incisions that you would make. And, and of course I can't do all the procedure. I only know the basics, and, and I wanted to include just enough for my students, but I included some so that his residents could do it. And I taught them how to draw the thyroid, and we're going to do that too. So we'll go back to that picture so we can take a look at that. And then we had the paper practice surgery. And what I did was I provided them with lots of different patients, these paper patients. So you could use the pencil as a scalpel, and that you could do the basics, like make this transverse incision, which is like, it's 3 to 4 centimeters above the sternum. And you could do that with a pencil. And then you have to reflect these muscles like the platysma and the thyroid and sternocleidomastoid and the omohyoid, and so that you can get to, through the fascia, the layers of fascia that they're gradated on, so that you could get to the trachea and you could get to the thyroid gland, which would be underneath this. And these are just 3 steps. I've got many others. And then because this is a total thyroidectomy, you would remove the thyroid gland, and then you would take up the parathyroids and be very careful with them, because that's the other really important thing. And you chop them up and implant them, I don't know, in the deltoid, and then you check the patient, and then you suture them up. So, I mean, it's not something, it's more than my students needed to know, but I thought it made a great way to make this actually work. So we're going to actually try this out a little bit. So I hope everybody's got that paper with them. So if you've got the paper, I'm going to go to these last questions before we go to the paper. So you might ask yourself, when you're writing something like this, how does evolutionary thinking impact the way we visualize the body and an organ or gland in this case? And it's a follicular gland, and what does a follicular gland feel like, and what does that mean? How does drawing help us think about all these topics? And if you have any thoughts and you want to share them, you can do that as well. And then I'm going to stop this, and we'll go to the other screen. Let's see, where did I put my Krita thing? So I can. Pardon me, I think I may have this. There we go. Okay, now I can do it, I think. Wait a minute, sorry about that. I'm confusing myself here. Oh, there it is. Okay, we'll share that. Okay, so I have a drawing under here. Here it is. So here's the paper practice guide that everybody should have a copy of. And like I said, it takes a while to do this. The idea was for residents to go through the surgery, or medical students. My students just did a little bit of it for fun. And I would draw in some of the lymph nodes, because if it's a total thyroidectomy, you might also take out the lymph nodes. So I put them in there, and they're in different little areas. Remove these, and that's like I saw them doing this in the surgery, so I said, well, maybe you should put the lymph nodes in there, especially if it's probably cancer, Graves' disease, or something, most likely cancer of the thyroid, and you put those in there. And the other thing you want to put to sort of anchor your drawing is the aortic, the arch of the aorta. So you can just make a cylinder like this, make a cylinder like that. Put your branches off of it. Just, you don't have to make it perfect, just so that you have the branches. And you just have these branches here. And that gives you some thinking about this, like what might be in the vicinity of what you're doing. Got these, I mean, I'm not going to draw all the vessels in here and all the nerves, because I think, when you look at a posterior view of the thyroid, you've got this right laryngeal, nerve going around, looping around some part of the, I think, the subclavian artery, and that's a more complicated thing than we want to do. So we've got this basically in here, and we've got our aorta, and we can give a little darkness around it to make it stand out a little, and you can do that on your paper too. So we can make our aorta stand out. So over here, and we know the sternum is in that vicinity. So what we're going to do is with our pencil, and I could make a different color, but I won't. You should use a different color, I guess. We're going to make our transverse incision right around here. So we would draw just a line, and then we would draw the scalpel coming in and helping us make that incision here. So we would do that, and that would be our first step. And you'd say, oh, 3 to 4 centimeters above the sternum, and you write that in there. Now, I provided them with a bunch of pictures because, obviously you can't draw all this stuff on one picture. So I'll just give you an idea. So then you start to think about the hand positions and the incision. You want to make it small, and the strap tension lines, and you make some skin scratches and the platysma muscles there, and you deepen the incision, and you start to open this flap up. So you would open it up. You'd start to see this flap opening here. And you'd start to see some fat, some tissue in here. And so that would be like the beginning of what you'd be doing on your paper. Now, like I said, I'm going to jump ahead, but this would be, you'd open this up really wide and you'd stretch this up like this. And we'll erase some of that so you can stretch it up with that. And you see these kind of like tweezers here holding this back and holding this back, and you can erase the scalpel and hold back here. And then you're opening this up so you can use the pencil again, and you can deepen the incision, and you'd see the platysma muscle here. That's the one like when you make your face, kind of like upside down grimacing, that would be there. And I wanted my students to know that you would, that your pencil would function as both a sharp and blunt dissection instrument. So you be careful when you're cutting things in the planes you're cutting them on. And I'm going to see if I can get a color here and change this a little more. We'll go a little more deeply into it. That was easy to draw. I don't think that was hard for anybody. But we would see the muscle being pulled back here, and because we'll put a retractor on it. We see another muscle being pulled back here. And then we see a muscle underneath it. So these different planes, fascia reveal these different muscles. And so we can put where am I blacking up? We can put our retractor in here. So we'll it's still turning, sorry, trying to get black here. There we go. And we'll put that in here. So we have a retractor here. We have a retractor here. And like I said, it takes a couple of pages to do the whole thing. But there's our retractor, and we've pulled back a little bit. And we've got we're going to go through these muscles as well. So you just put them in. And of course, when you're learning this, you start memorizing the muscles, and you start drawing them and memorizing them at the same time, which is really helpful because you're going through this step by step and slowly. So now you've got this feeling, and then you start to see a little bit of the thyroid under here. You start to see a little of that. So you see like a little bit of the gland, you get that in there, and we've deepened it. And you start to see that you're getting to where you wanted to get to. And now you have a little advanced feeling of what you're doing. And then you open it up even more. And so we make a line down here, and we'll pull this back even more. And now you're deeper into this tissue. And now what you're going to see is that thyroid gland and the trachea. So you're going to see the trachea, and it's going to look like this. And of course, you start the trachea, all you need is a cylinder like this. This is going to be your trachea. And then you're going to remember there's C-shaped cartilages in it, and they have connective tissue in the back, so they're flexible. And you would have these rings like this, right? So that would be your trachea. So you know that that's going to be right here. So you'll see that. And then you'll see the thyroid gland, which is that butterfly-shaped gland. And you'll see it look like this. You'll have like an isthmus, if you say that, and you've got two lobes there. And you can draw a little bit of like this to make it look glandular. And so what you would do is you look for the recurrent laryngeal nerves, you look for the important vessels, and then you start inspecting the lobes. You're going to remove them, and you free them, the fascia, from because the thyroid cartilage is up here and the cricoid cartilage is there, but you would see those cartilages here, and it might be good for you to draw those in. You can remember them. There's a thyroid cartilage here and a cricoid cartilage below it. And so now you have the thyroid and you want to remove it. So you would probably put your finger under it and palpate it a little, pull it out, and then you snip it away or you cauterize it away because it's very, very vascular. And then you'd be left with, I'm not going to draw the in-between parts because this is in the book, but like I said, it would take a little bit. So you'd be left with just, you'd see the trachea, that would be a total thyroidectomy. And you'd see some fat around there and some fascia and tissue. And there you'd still have the retractors on it. And then of course, the last step in this procedure is to close the patient back up. So these would be leaving you and this would be leaving you, and we get rid of this. You'd work on another paper and you'd see that this incision you made here is going to start to close up. So you know, your hands would be on here. You'd have gloved hands and you'd be in position. Your hand would be over his face. And then ultimately the incision would be sutured. And your procedure would be done. So, you know, I made a model of this got a dowel and I built a clay thyroid around this. And I think that's always a good idea to make your own model. So I made my own model. And of course, you know, from the back, if you looked at it from the back, it's kind of neat because you think like the gland actually wraps around the trachea and looks kind of like this from the back. And then you see the superior parathyroid and you see the inferior parathyroid, and you start to think about this, not just from the anterior view, which is what most students think about these things as. They have a totally different view when you look at it this way, and you make models and you rotate things. And so there's your patient. Your patient is alive and well and finished. Nobody got hurt, and you got to experience what it would be like to go through this surgery. But most of all, what you got to experience were the terms and the ideas that students just rush through them. Like, I'll memorize this for the test and I'll be done with it. And they don't really get a feel for it. So this book was a lot of fun for me. It was a lot of work, but it was a lot of fun to use in the basic anatomy. I did it with high school students too, and they were perfectly into it. They liked the whole idea. So I'm going to stop there and ask if anybody has any questions. And that's our, you know, my last topic for the night. So I'm sure everyone's asleep by now. If you want me to draw the, you know, sleep-wake cycle, we'll do that. You can go off to bed. And thank you for having me. And I hope you enjoyed this activity and the experience and got a taste of how all these things are different but interconnected, and that maybe you'll take up some sketching and drawing.
Karen, thank you so much for that. That was just absolutely stunning. We have 2 or 3 questions. We have a bunch of questions, but Aditya, what you got?
Yeah, so first off, the first one will be, when you encounter a concept with a lot of information or events that need to be addressed in a visual presentation, are there strategies that you use to organize the information?
Yes, I think when there's a lot of information and you're not sure where to start, think about how what the core idea is. I mean, if you're talking about like a lot of genes interacting with each other, how would that look in your mind? And what's the main word that would represent this? Now, sometimes I make lists of words that I think represent ideas that I want to draw. And then you play with it and you play with it. And then eventually it starts to build on itself. So find a core idea that represented, like maybe networks is the core idea, like epigenetics. A lot of people refer to Waddington and this drawing he made of like the epigenetic landscape. And a lot of people are using the word landscape now. So what word would represent your big idea? And then from that, build your small ideas. That's what I would do.
Do you have,
it overwhelms us. Yeah.
Do you have any resources that you'd recommend to beginners or intermediate artists?
I think, and this is a lot of people think just like with instruments, if you get like a Les Paul guitar or, whatever, and you get the best drawing book, that somehow you'll learn from it. The best thing to do is draw from life and sit with a sketchbook wherever you can and struggle with it. And once you do that, then you can pick up, there are a couple good anatomy books, anatomy drawing books. I'm trying to think of the method right now. I can't off the top of my head, but Brigman's. I think Brigman's drawing is a really good book. But a lot of people get these books and they buy a lot of art supplies and they don't know what to do with it because it's like, oh, this is exciting. Then they get it and it's like, oh, this is work. And then they let it sit there and everything. It's like, look at my shiny yellow Les Paul, but I don't know. Play that. It's too much work. So I think play around with it, make lots of mistakes. Because one thing you'll never notice, despite there being the great masters, no one's ever found their mistakes. Isn't that funny? Like they probably threw that stuff away. So you can make as many mistakes as you want. And then your body starts to get acclimated to that skill. And it doesn't have to be great. It doesn't have to be a great skill. It's just for your purposes. So I would say get yourself a sketchbook, make some storyboards that are empty and try and fill them in. Try and fill in like a comic strip. Try and come up with an idea in a storyboard with like 5 frames in it to describe something like the microbiome and see if you can do it. Challenge yourself. One thing I could say is stay off the internet a lot, as much as possible, because the more visual stuff you see, and we don't even know it's happening, it hijacks your imagination. Everyone's vying for your attention, right? These commercials are coming at you and they're sticking there subconsciously. And the more you avoid it, the more you put your phone down, the more you sit in real time and space out somewhere watching people or sitting in nature, whatever, or ocean waves, the more you get an imagination. When somebody hijacks your imagination, then you're always going to look to somebody else's idea, and it makes your brain lazy. So that's what I would say. Just less time. We're all guilty of scrolling through millions of videos of people doing things like jumping off of cliffs and stuff like that. But I mean, that's just time wasting. And you start to gravitate away from that, and you start to find your own skills much more satisfying. Entertain yourself. See if you can do that. That's what I would say.
And we have one final question. Many of us students grew up watching Magic School Bus. I was wondering if you were familiar with this and if you were influenced at all by that show or what you thought of it.
Well, you grew up watching Magic School Bus and I grew up watching the Banana Splits. So I'm familiar with the one where they went through the intestine, and that's kind of like Fantastic Voyage with Raquel Welch and all these other people, where they went into the body. And then there was a remake of that, like in the 1990s, I think, with Martin Short. He did another thing where they also went into the body. And I think that perspective is great. And I think it does influence you. You don't want it to influence you too much, but I would always be asking myself, what would it really look like in here? If I could see what was here, would it be in color? Would it actually have a tangibility to it? Could I touch it? Or is that phenotype, that the transient state that is like in a sort of a fuzziness that I can't really feel? And I think it would be more like that. I don't think it would be this solid imagery that we are presented with. But certainly shifting your perspective, points of entry. I mean, I always think the anthropomorphic is good. I always think the animal's perspective is important because a lot of times in biology, we dismiss other living things as not being intelligent. just being around other living things, I know they're way smarter than me. I mean, and I'm really impressed. I think that humility is your best, I think one of your best skills to have, because then you really opens up a lot for you. When you think you know everything, then it's all done. So I think that's the best thing to do. But Magic School Bus is cool. It's a good thing to watch. Cartoons are very good. Watch a lot of cartoons. The older ones are better. So, Three Stooges aren't bad either.
Well, thank you so much for answering those questions. I'm going to share my screen real quickly just so I can share the quiz information with the student.
Sure. And I'm going to stop this and go.
Okay, can everyone see my screen?
Yes.
Okay, I'm also going to put into the chat all of the links to the quiz for today's session, as well as session 27, which is our reopened session for this week. So upon completing your session quiz for this session, session 103, with a 70% or higher, you will be prompted to download the certificate. Please make sure you do so and save it for your record of attendance. In the chat, I've also put in the session 27 quiz and video link. There will be no due date on that quiz, so please be sure to copy that down for yourselves. Dr. Fowler, do you have closing remarks?
Karen, that was just wonderful. Thank you so much. Can everybody online, all 200 of you, please put thank you, Karen. So look at your chat and watch these pop through. It's kind of cool.
Wow. Well, I'm sorry this went so long. I thought I'm going to really cut this down so it's short, and then it ended up really extending itself. Thank everybody for coming, and thank you for being so patient going through this and looking at it. And I hope it, made you so tired, you're relaxed now, and you can go to bed. So,
well, Karen, we're going to put your talk online. Probably 5,000 people will watch it online. Each one of them is a future healthcare provider, and we'll see probably 100,000 patients in a career. 5,000 times 100,000 is a 500 million. Wow. So tonight, Karen, you have touched 500 million lives in the future. So just thank you so much.
Oh, thank you for giving me the opportunity to talk about something that, a lot of people don't talk about, but is a lot of fun. And I hope there's some inspiration in it for anybody watching. And I appreciate the experience, and thank you everybody for coming. Look up those people. They're really cool, the historical people, especially if you're into medicine, and you'll find a whole other world, give you lots of ideas. So I guess I'll let you guys go for tonight, right? And I appreciate everything. Thank you very much. Have a great evening, and good luck with this magnificent program you've created, and wish everyone well. So take care.
Well, thank you, Karen. And so on behalf of our wonderful speaker tonight and the whole virtual shadowing working group, we want to thank you for coming tonight, and we wish you a good evening